everyone, this is Miss Chiao. We're in the carbon unit. This is the Bromothymol Blue Lab. And the question we are trying to answer is, how does carbon dioxide affect water? We noticed when we played the carbon cycle game, there were two areas in which carbon traveled between the atmosphere and the surface ocean. So in the atmosphere it's the carbon dioxide, but when it goes into the water it becomes carbonic acid, which is a weak acid. And we're going to see how does the pH change and how eventually that pH does affect the living things that are living in the ocean. So let's go ahead and do safety first. You've got to have your goggles on, and that when you are pouring from your stock solution to prevent contamination, you always pour the bigger stock into the solution if you ever run out. Okay, let's go ahead and do our material list. You have a timing device that's set to, let's do 5 seconds, or you have a timing device on the watch for 5 seconds. Then you need a straw, 400 milliliter beaker for liquid waste, 100 milliliter graduated cylinder, 50 milliliter flask, 1 milliliter pipette, and a diluted bromothymol blue solution. Okay, let's go ahead and do the procedures. First, put on your goggles. 2, you're going to measure out 50 milliliters of bromothymol solution. You're going to use this first to pour, and we want to get to 50 or close to. Once we get to close to 50, so we don't go over, you're going to pause. You're going to next use 1 ml pipette to take some from here. You always take it up, we don't put it back in. And we're going to get close to 50 here. Want more? A little bit more. And when you're looking at the totals, what you want to do is you want to get it at eye level. So we have to go at eye level around 50, and it's right at 50 at the bottom of the meniscus. I may have some stuff left in here, we'll just leave it be. Pour it into your flask, and then you're set to do your experiment. We're going to add on in increments of 5 seconds of blowing. So one of the people, so there should be 4 people in the lab, split into 2 pairs. Each pair, that means 2 people, you're going to have 2 jobs. One person is going to be blowing the solution, and you got to blow very gently. The second person is going to be a timer. Since I do not have a second person, I'm going to count to myself 5 seconds while I'm blowing. Now it's very important that you blow gently. If you don't, the water will come back up. So, oh yeah, I have a mask on, I forgot about that. So, okay. That was 5 seconds, and then I'm going to note the color. How do I know the color has changed? You're going to compare it to your solution from before. Okay. Ideally, you want a solution to compare to with another group. So you have to figure it out, write down the color, compare it to the color that's on your lab paper, and then you're going to keep on doing it. Do another 5 seconds for the second one. And some of it splashed out, and you notice the color has changed again. So note the color, note the pH, keep on doing it until you get to the color yellow or close to the color. So blowing 5 seconds each time, and note that. All right, and then for cleanup, what you're gonna do is that you're gonna rinse this off because you've been blowing through it, has your saliva in it, is that you're gonna dump this into the waste. You're going to rinse these 2 with water. So I'm going to go rinse. Once they have been rinsed with water, this is ready to go for the next group. And what you're gonna do with the scissors is you're gonna stick it in here and cut all the way through so that they don't get stuck in animals out there. Okay? And that way it's ready to be thrown away. Make sure this area, if you blow the bubbles and there's little spots, all you do is grab a paper towel, wipe it down. Oh, we do have the spray bottles. So we have cleaning solution. And once you're clean, you're going to go and wash your hands, okay? That's it.